Okay, so as I promised, we're moving into kind of a completely different area of Bio 110, at least relative to the last few lectures, last few modules that we've had, which have been largely genetic, molecular genetics, etc. And there is a connection. It's not um, quite as strong. It's actually, this is going to be content that most of you will probably be seeing again in greater detail when you take Bio 230, which is microbiology. Um, I have no problem with giving you a little bit of a preview. It's actually kind of an important point for you to have uh, this foundation before you move any forward. Some of you won't be taking micro for a couple of semesters. So you can sort of think of this as a little bit of a taxonomic survey of a whole bunch of things we might call nasty microbes, and things that you might be studying in a clinical microbiology class. Also, you call it, as I've pointed out here, all the nasties that can make you sick. And we'll start with viruses. And viruses is it's a topic that's covered in several chapters, actually several different points in the textbook. I've just selected a few kind of, I realize that they're all over the place as far as what chapters they're in. But since this is a little bit of a survey, we're not really delving deeply into any of these taxonomic groups. I just really, at this point, need you to understand what a virus is, how it's different from a bacterium, how both viruses and bacteria are different from the other kinds of agents, parasites that might cause you to be sick that are from a eukaryotic source. And so let's go ahead and move into this. We'll start with viruses. In fact, I'll point you in the direction of chapter 10, section 18. Let's see if I could find that. Shares. Here we are. Again, there are several different places you can go to get this information. The main concept here is that with viruses, we really don't have any parts of a cell apart from some very rudimentary things that make it, mildly biologically similar. I mean, you have a genome, of RNA genome, like what's shown in this diagram. You could also have a DNA genome, right? There is a coat made of protein, and sometimes you actually have a membrane-like structure. It's called a viral envelope, and the viral envelope is something that's taken away from the host cell that it was infected from the previous turn. Okay. And the basic idea here is that you have a viral particle, right? Let's say a viral particle. Sometimes you could call it a virion, which is the thing that gets transmitted from cell to cell. Okay, it's also the thing that gets transmitted from host to host. And so when somebody sneezes and you catch a disease, a viral disease, as a result of encountering that sneeze, what you are encountering are the viral particles, right? Now, once the viral particles get into your cells will start releasing viral particles of the same type, and they will go off and infect other cells in your body, right? So you've got infection from a different host into your cells, and you've got infection from your cells to other cells in your body. And every time that's happening, there's an infection of a host cell. Which is really crucial for the virus because it's how we basically make more viral particles, right? So the idea here is that the viral particle gets into the host cell. The host cell is used as a little factory to make more viral particles. And at the end of the process, we end up with lots and lots of viral particles. If I could scroll this down. Okay. we have exiting not just one viral particle, but um, millions of viral particles. I mean, if it were just one virus coming in, one virus leaving, then there would be no big deal. Okay, but because what's happening here is the whole host cell is being taken over, it's converted into a viral machine, which makes more of these infectious particles, which can then go out and infect more of your cells, which can then produce another millions of viruses each one, which can then go out and infect other cells. I mean, you can see how this is going to very rapidly cause potentially a lot of damage, given the fact that the cell death is often an outcome as a result of having been infected by the virus, having been used to make tons and tons of new viral particles. Oftentimes, cell death is the reason why viruses make you sick, right? Yeah, the viruses, all they're doing is making more viruses. But in the process, they're taking over your cells, commandeering the cells by chemical machinery, making more viral particles, and then the cells are pretty much toast after they've been used and abused in that way, right? Yeah, so viruses are doing things kind of selfishly. They're making more viral particles. The host is generally damaged because of the cell damage is caused resulting from it. One thing we don't see very frequently in viral diseases is the production of some type of toxin. We're not, we're not intoxicating the cells. That's a really common theme in bacterial diseases. Bacterial will often produce toxins which will cause damage to the host. That's not nearly as much of a factor in the case of viruses, right? The other thing here is that in the cycle, what's happening is that we're getting lots and lots, we're taking the viral genome, okay, making lots of new viral genomes over here. We're making more viral proteins over here, and then we're assembling, right? It's like we're deconstructing the virus into protein or into the genome, and then we're reassembling the new particles. And so another thing that's happening that's different between viruses and bacteria is that all bacteria are cells, and a bacterium is going to have to undergo cell division, and we'll end up producing two cells, right? We call this binary fission. Okay, viruses don't do this, right? Okay, this is a characteristic of cells. When viruses reproduce, they're always undergoing this deconstruction. We're going to be uncoding and releasing the viral genome. And then the viral genome is then going to make lots of copies of viral protein, lots of copies of viral genomes. We're going to be replicating the genome. And then once we have all the proteins and the new viral genome, we're going to assemble a new generation of viruses, which will be there in the millions. And in the process, we're going to cause the death of the host cell that we've infected. Okay. So that's viruses, and I've got a whole thing with my other share on the whiteboard, which illustrates this one more time. Okay, and I'm doing this again because I want to point out a couple of things that will be important later on in this module when we start talking about the immune system, because well, I mean, there's a little bit more detail, right? Um, uh, viral particle, or the virion, okay, consists of a viral genome, okay? And then we talked about the protein capsid. Now, the protein capsid is going to come in a lot of different shapes. The two most common regular shapes are this icosahedron. It's like a 20-sided die, right? That you roll when you're playing D&D. But in this case, the icosahedron is basically made up of triangular panels of protein, and the protein has little markers that are little joints at the very tips. And these protein, sometimes we call them spikes, are important because they help uh, the viral particle get into and uncoat inside of the host cell, right? They're also important because they are potential markers. These are things that we can use to identify a virus, okay? Because these protein spikes that are there in the virus are going to be different from any of the proteins that we find inside of a normal host, right? And so, yeah, I mean, when we look at, when we as biologists look at viruses, we're always on the lookout for things that can uniquely identify either a viral particle or a virally infected host cell. Because if we were to do something to either destroy the viral particles or to take those cells of our own that are infected by viruses, and we could kill them, it's right, kill your own cells before the cells have a chance to produce the next generation of viruses. That's going to tremendously help our ability to recover from the viral disease, right? And so, yeah, viruses is this molecular breakdown of the viruses into the protein capsid versus the genome. Yeah, we're not really going to be creating immune defenses against the genome so much, but we will be looking specifically at those markers, those protein markers, like the protein spikes on the capsid, or like, as was in the picture that we showed you from the textbook, these spikes that are occurring on the envelope. Okay, because sometimes a virus is going to have a membrane, a phospholipid bilayer envelope, which is a little piece of membrane that it's taking from the infected host cell. Right? And so, yeah, the general picture here is that this viral particle comes in, either it will come in as an envelope virus, or a naked virus, one that doesn't have an envelope. And what gets inside, what we eventually get inside is either the viral genome directly as a result of uncoating, because this protein capsid will squish out the viral genome onto the inside, or it's going to meld with this and we'll get the whole capsid inside. And then the whole capsid will then release viral genome, right? At which point the viral spikes are going to be out here on the outside. And this is what I meant. This is what I meant by the way that we can actually use proteins that are on the surface of an infected host cell to recognize a host cell that could be marked for destruction, right? These envelope proteins are going to be there when the initial virus goes in. It's also going to be there on the way out because before we actually make the next generation of viruses, we need to put a whole bunch of envelope proteins on the outside of the membrane so that when the virus comes in and takes in, when the new viral particle comes in and takes away a little piece of the membrane, those viral spike proteins are going to be there, right? So in other words, these viral proteins are going to be there on an infected host, but they're not going to be there on any host cell that has not been infected by the viral parasite, viral agent. Okay. Yeah, and then what happens after that is we make lots and lots of copies of the viral genome, make lots and lots of copies of we'll undergo replication and then translation, and we'll make lots and lots of proteins, viral proteins, and then we'll make more of these viral capsids using the viral proteins. We'll put the viral genome on the inside, okay, and then we'll produce tons and tons of viruses, and that's pretty much how things work. Thing that we're going to be looking at here, okay, is that the viral particles themselves are recognizable to immune defenses because of their unique viral proteins. Okay. I'm sure you've heard about the spike proteins on SARS-CoV-2, right? You know, these used to be a lot more obscure concepts, but now it's everybody and their um, dogs are talking about microbiology, virology. Um, it's not so much an unusual thing for people to know something about viral spikes. And when we say viral spikes, we're referring to the proteins that are there on the surface of the virus, either on a naked virus or these envelope proteins in an envelope virus. And they allow us to recognize both the viral particles as well as the host cells that have been infected by the viruses. So yeah, kill off either these guys or kill off the viral infected host, and that would be a happy day for the infected host himself. Okay. Now, when I talked to we've pretty much covered everything here. You know that they're not cellular, they're not alive because in order to be a living cell, must be cellular. Okay, so just telling you that they're not cellular is also telling you that they're not really living. Okay, we talked about the viral replication cycle and the way that the, the viruses are kind of vulnerable because their proteins are different from the ones that we have in our cells, and we're targeting those proteins with our immune defenses. Okay, now on to bacteria. You know enough about bacteria at this point. We've been talking about them in the context of like the lac operon and the trip operon. These are things that are a little bit different. They're not, the operon thing is not something we can really target. There are some more obvious targets. And the reason why we need to target bacteria is because bacteria sometimes they cause disease, right? And so I've pointed out a couple of points in the textbook where you can go look at the characteristics of bacteria and some features about them that make them particularly challenging as far as agents of disease. But as things go, they're really kind of help us destroy them because they are so different from our cells, right? So now we're thinking about bacteria. Remember, they're prokaryotic, and because they're prokaryotic, they're going to have some key differences with our cells, with human cells, right? Because we are eukaryotes and they are prokaryotes, and specifically they're bacteria. Just remember, prokaryotes include the archaea as well as the bacteria, and the archaea are a little bit different. The archaea are actually a little bit more similar to us. But since most of the things that cause disease are prokaryotic will be bacteria, we can look at these key differences. Specifically, and in particular, there are two that are of great importance. One has to do with ribosome structure. Eukaryotic ribosomes are 80S, and prokaryotic ribosomes are 70S, which means that they're about half the size. I mean, yeah, they're really much smaller compared to the 80S ribosomes of eukaryotes, and being so small, being so structurally different from eukaryotic ribosomes, we can do things. We can put poisons in that will target ribosomes that are 70S, but have effectively no chance of harming 80S ribosomes functions, right? And so when you look at some kinds of antibacterial drugs, erythromycin, chloramphenicol, different kinds of tetracycline, there are lots of different drugs that are related to these things. There's oxytetracycline, there are different kinds of chloramphenikols. There's a whole class of drugs called macrolides, which are basically erythromycin variations and erythromycin derivatives. All of these things are going to be making 70S ribosomes do things that they're not supposed to do, right? They cause malfunctions in 70S ribosomes, which is basically going to make the cell's protein synthesis it will disrupt protein synthesis, making the cells unable to divide, right? And so that's going to be an important way that uh, we can target. See what we're doing? We're looking at us, we're looking at them, we're saying, hey, 70S ribosomes is one way that we can specifically target bacteria without having any collateral damage to our own cells, right? And so this this use of differences between them and us is an important feature of bacteria that we make great use of, right? Um, Another one is the fact that bacteria always have peptidoglycan in their cell walls. And peptidoglycan is a very unusual substance. There are a couple of monosaccharides that exist in peptidoglycan that don't exist in any eukaryotic cells. And we could target that. Okay, penicillin and all the other psyllin drugs like ampicillin, methicillin, amoxicillin, all the psyllin drugs are related to penicillin. And what they do is they go out and target the synthesis of peptidoglycan. Okay? And if the cells are not able to make new peptidoglycan properly, they're not able to make cell walls properly. And so these guys are going to be targeting cell wall synthesis, which is going to effectively make it impossible for the bacterial cells to keep their cells together and they fall apart and die, right? And so yeah, these are all really good things. These are drugs that have been known about since the early 1900s. They've become more diverse, better, less dangerous over the course of the 1900s. And by the time we got to the end of the 1900s, we started getting rather concerned because these drugs stopped working. Not You didn't have to wait all the way till the end of the 1900s. By the time we got to 10, 15 years after the introduction of penicillin, we started seeing penicillin-resistant strains of bacteria. Okay. And I guess my point here is that bacteria evolve. Okay. For every drug that we throw at them, right, throw erythromycin at a bacterial cell, the bacteria will be affected. They'll be harmed dramatically by the erythromycin, but a few of them may survive. And those that survive will be resistant, and those that are resistant will make a whole new generation of bacteria that are all slightly resistant to erythromycin, right? And then the cycle begins. We've got a whole bunch of cells that are a little bit more resistant to erythromycin, and then we subject them to more erythromycin at a higher dose. And yeah, most of those cells are killed, but a few of them survive, and we end up forming a whole new generation of bacteria that is even more resistant to erythromycin. And the cycle continues. And basically, it's the problem of administering a drug that is at too low of a dose to make a 100% kill, right? Because if we had completely wiped out the cells with the original dosage of erythromycin, then there would not be any survivors to promote higher degrees of erythromycin resistance in the next generation, right? But we don't always do things correctly. And so when we use low doses of tetracycline in cattle feed to make the cattle grow faster because they're not wasting a lot of resources feeding bacteria, we're basically exposing the bacteria to low doses of tetracycline and causing them to evolve higher degrees of resistance, right? When we're administered a dose of cephalosporin because we have strep throat, right? And we take our cephalosporin, but after like one day we feel a whole lot better and say, hey, you know what? I'm not going to finish my cephalosporin course. I'm just going to be happy with the improvement that I have. Yeah, that's not a good thing because those cells that happen not to be killed because I didn't administer enough of the drugs to get that 100% kill, those few survivors are going to be coming back and their offspring are going to be a little bit more resistant to cephalosporins. Okay? And so the key concept here that are associated with bacterial diseases, when you first, they're not viruses. They are cells, right? And they have ribosomes, they've got cell walls and all the like. And we're happy because we've got these differences, but we also know that there are mechanisms that can allow them to become resistant to the drugs that we throw at them, right? Uh, the, uh, the bacteria can evolve enzymes that will come and just basically snip apart these psyllin class drugs that we might be using, right? So if I'm a bacterium and I produce a penicillin destroying enzyme, then the penicillin that you take against me, the bacterium, is not going to be very effective. Right? If I develop a way of causing the cells to eliminate tetracyclines before they have a chance to um, affect me, right? That's going to make me resistant to tetracycline because those drugs will never have a chance to affect my ribosomes, right? So, I mean, there are lots of ways for bacteria to become resistant, tolerant of these drugs. Pretty much everyone has happened. And this is the state that we're in right now. This is what you need to know about bacteria. Okay? But important thing here is that they are different. Okay? Technically, apart from these resistances that have evolved as a result of our practices of simply using the antibacterials, right? Um, apart from that, the fact is that they're actually different from us, and therefore they're easier to treat compared to things like uh, parasites that are eukaryotic, right? Remember eukaryotic? That means they have the same kind of cells that we do. They've got envelope nuclei, nuclear envelopes. They've got endoplasmic reticula. They've got Golgi apparatus. They've got mitochondria. They've got no, they don't all have chloroplasts. If we're talking about algae, they would have chloroplasts. But parasites that are eukaryotic, first of all, we call them parasites if they're eukaryotic. We generally don't call bacteria parasites. We call them pathogens. Uh, viruses, we call them viral agents or sometimes pathogens. But we generally reserve the term parasites for those microbes that are eukaryotic in nature, right? And among these parasites that are eukaryotic, they might be fungal, okay? Fungi basically means they're more closely related to a mushroom than they are to either you or to anything else that's eukaryotic, right? And an example here would be something like a yeast, Candida albicans, thing that causes yeast infections. Yeah, a yeast infection by Candida, okay, is caused by a eukaryote, which means that inherently the antibacterial drugs won't work. Because those antibacterial drugs are targeting those things like 70S ribosomes and peptidoglycan synthesis that are not going to be there on Candida because Candida is, after all, a eukaryote, right? Uh, there are animals, in particular helminths, right? Which are also like worms. You can look in the textbook for pictures of nasty uh, nematode worms that are there inside of the heart of an infected, I don't remember what it was. Maybe it was a goat or a cow, right? And scary pictures, okay? But worms are not going to be affected by antibacterials because worms are eukaryotic, right? Now there are drugs that are specifically antifungal, right? Like uh, clotrimazole. Clotrimazole is one of a few different kinds of antifungals that you might be using if you're actually experiencing a yeast infection. Okay. There are anti-helminthic anti-helminthic drugs like ivermectin. I don't know if you were following this during the bad times of the COVID epidemic. Ivermectin is something that you give to horses when they've got worms, right? It's not an antiviral. And there's absolutely no reason why anybody should expect there to be any beneficial effects of an anti-helminthic drug, which is specifically targeting worm animal parasites in the case of, the whole thing about ivermectin being a therapy for COVID or a preventative thing was completely ludicrous, okay? And somehow it gained some steam because some people were actually promoting it. And people believed the people that were promoting it. It was a little bit hilarious, a little bit sad. Okay, in any case, we do have animals, fungi, and there are also a whole bunch of other things, not animals, not fungi, not plants, because plants don't usually infect us, but if there were plants, they would not count as protists, but still eukaryotic. Okay. That are also going to be parasitic. An example of this would be malaria. Malaria is caused by a microbe called Plasmodium. Okay. That is, yes, transmitted from one host to another by mosquitoes. But it's not the mosquitoes that are causing malaria, right? Mosquitoes, they're not nice things, but they don't cause malaria. The cause of malaria is this microbe, okay? Which is why it's not even accurate to say that mosquitoes kill more people than crocodiles, because the crocodile, I don't know if you've seen that graphic where, what is the most dangerous animal in the world? Is it sharks? Is it hippopotamus? Is it crocodiles? No, it's mosquitoes because there are more people that die of malaria. No, that doesn't work because mosquitoes don't cause malaria. Mosquitoes carry, that is the organism that causes malaria, but the correct answer to what causes malaria is different species of Plasmodium, which has several, there are 4 species, Plasmodium vivax. I can't remember the other ones, but vivax is the one that I remember. Falciparum. And none of these are actually mosquitoes. They are things that kind of look like this, right? Okay. They have a little apical complex. And there are cells that look like this, and they will infect red blood cells, and they will cause you to get malaria. But the actual organism is a microbe, right? And like fungi, like animals, we cannot use antibacterials to treat them because antibacterials will not work. Okay? So the downside of these is that all these drugs, the things that cause the antimalarial drugs, the antifungals, the anti-helminths, the drugs are going to be a lot more likely to cause damage to your cells because your cells are physiologically more similar to the parasites, right? Because you're both eukaryotic, right? And so, again, coming back to the one of the focal objectives for this particular lesson, one is to kind of set us up in our discussion of immune function. Which is where we're moving in the next lectures, but also as kind of like a backdrop for the things that you'll be thinking about as the microbes that cause disease, which is an important bit of literacy for those of you who are going to be having some role in the health professions. Okay, yeah, if you have gotten to this point without having a clear idea of the difference between viral diseases and bacterial diseases and diseases that are caused by eukaryotic parasites, let that phase of your life end right now, and now you can have at least a little bit of an appreciation of the differences. Okay,